Dear students, after studying this module, you shall be able to learn about the basics about the use of supramolecular entities as molecular devices, understand the concept of energy and electron transfer processes, learn and analyze the term semiochemistry, know in detail about molecular and supramolecular devices as photonic, electronic and ionic. Now students, let us begin with the introduction of this topic, that is supramolecular devices. I would like to introduce first molecular devices. Molecular devices have been defined as structurally organized and functionally integrated chemical systems. They are based on specific components arranged in a suitable manner and may be built into supramolecular architectures. Thus, a supramolecular device is a complex system made up of molecular components with definite individual properties. These properties are intrinsic to a particular molecular component, whether it is part of the device or whether it is not a part of that particular device. Another way of saying this is that the interaction energy between the components of the supramolecular device must be small as compared with the energy, that is other energy parameters which are relevant to that system. Such a definition of molecular device does not exclude the traditional concept of host-guest or receptor and substrate. Molecular recognition events between host and guests may be an intrinsic part of the operation of the supramolecular device, which might, for example, be designed to bind and then signal the presence of a guest. The function performed by a device results from the integration of the elementary operations executed by the components. In other words, students, we can say that photonic, electronic or ionic devices, depending on whether the components are respectively photoactive, electroactive or ionoactive. That is, whether they operate with or accept or donate photons, electrons or ions respectively. This defines fields of molecular and supramolecular photonics, electronics and ionics respectively. Two basic types of components may be distinguished henceforth. First one will be the active components. Active components are the components which perform a given operation, that is acceptance or donation or the transference on photons, electrons or ions. Students, now the second type of component, that is the structural components. These components participate in the build-up of the supramolecular architecture and in the positioning of the active components, in particular through recognition processes. In addition, ancillary components may be introduced in order to modify or perturb the properties of the other two types of components, that is the active components and the structural components. A major requirement would be that these components and the devices that they bring about perform their functions at a molecular and a supramolecular level as distinct from the bulk material. Now students, I have introduced the topic of molecular devices and related it to supramolecular devices. Now let us go in detail of this topic and look at the topic of molecular recognition, information and signals semiochemistry in a detailed manner. Molecular recognition events represent the basis of information processing at the supramolecular level. They may give rise to changes in electronic, ionic, optical and conformational properties and thus translate themselves into the generation of a signal. by making use of the three-dimensional information, that is storage readout operating in molecular recognition in combination with substrate transformation and translocation, one may be able to design components for devices that would be capable of processing information and signals at the molecular and supramolecular levels. The discussed molecular recognition events can occur only if correct selective binding of the complementary active components takes place. Now this you can see in the figure, and these processes may play a role in several key steps, and these steps are the building up of the device from its components. Second step may be its incorporation into supramolecular arrays. Third is the selective operation on the given species, for example ions. Fourth is the response to external physical or chemical stimuli, which may include light, electrons, ions, molecules, etc., that may regulate the operation of the device and switch it on or off. Fifth would be the nature of the signals generated and of the signal conversion effective, that is the photon-photon, photon-electron, electron-electron, electron-ion and ion-ion, etc. Recognition dependent informed devices represent the molecular and supramolecular entities by which molecular recognition events may be transduced into processes and signals through the design of suitable components responding to external stimuli and suitable for incorporation into the final superstructure. Recognition expressed via a chemical transformation in the nature or in the rate of formation of products amounts to the generation of a specific molecular signal, and the resulting area of investigation has been termed as semiochemistry, and this is obtained from the Greek term semion, which means sign or a signal. Semiochemistry is the name given to the area of supramolecular chemistry, which is broadly concerned with signaling devices. The term comes from semiotics, meaning the study of signs or symbols and their use or interpretation. The basic concept of a molecular sensor is that the substrate, that is the guest or the analyte, may be attracted to a receptor portion of the sensor. This is clearly represented in this is a straightforward molecular recognition event, and the receptor can be of any of the systems such as crown ethers, cryptands, cavitands, etc. The binding must be selective for the target substrate in the presence of a range of other potential guest species, depending on the system and its environment. In endoreceptors, the binding information sites are oriented into a molecular concavity, whereas in exoreceptors with outward directed sites, present informed surfaces on the recognition may occur. The receptor must also be in communication with a signaling unit that is responsive to the guest binding, which generates a signal in the form of an emission of electromagnetic radiation, that is photochemical sensing, a current, that is electrochemical sensing, or an otherwise externally measurable change, for example in color or in pH. Now you can see this figure, it is showing you the model representation of a chemical sensor that occurs in the molecular recognition event. Now students, let us come on to the topic of supramolecular photochemistry, that is recognizing and functioning in the molecular and supramolecular photonic devices. Supramolecular photochemistry involves the use of photochemically active components as the supramolecular devices and the formation of supramolecular entities from photoactive components may be expected to perturb the ground state and excited state properties of the individual species, for example, or that is the chromophores, giving rise to novel properties such as energy migration, photoinduced charge separation, perturbations of optical transitions and polarizabilities, modification of ground and excited state redox potentials, photoregulation of binding properties, selective photochemical reactivity, etc. When a molecular chromophore is irradiated with electromagnetic radiation of a wavelength corresponding to the energy required to promote an electron to an accessible electronic excited state, energy is absorbed resulting in the promotion of an electron from the ground state molecular orbital to the one having a higher energy. This is known as primary charge separation, and it results in a high energy electron and a positively charged hole. The energy of the excited state electron can either be dissipated as heat by relaxation by the solvent, that is called as non-radiative decay. It can be emitted radiatively, which is also known by the term of luminescence, or can be used to carry out a chemical reduction. Now this is explained in figure. Luminescence, this effect involving direct radiative decay in which the electron returns immediately to the ground state from the singlet excited state, is termed as fluorescence. Fluorescent emissions are usually of lower energy than the absorbed energy because the electron is promoted into a vibrationally excited state from which it relaxes non-radiatively before fluorescing back to the electronic ground state. Now students, here you can see the possible radiative events following the photoexcitation, and ISC stands for the intersystem crossing. Here you will notice both the luminescence as well as the fluorescence processes in this figure. Phosphorescence is a process in which the electron undergoes a change of spin state, that is intersystem crossing, to a triplet state. Then the triplet excited state, which is long lived, may undergo vibrational relaxation to subsequent lower levels or the lower energy levels, emitting a kind of luminescence. Fluorescent and phosphorescent processes may be distinguished by time resolved spectroscopic measurements because phosphorescence takes a longer time to decay than fluorescence. Now I hope you have understood both luminescence, fluorescence as well as fluorescence and phosphorescence. The result of photoexcitation may be divided into three broad categories. First one is the re-emission of the absorbed energy as light, that is by the process of fluorescence or phosphorescence. Second is the chemical reaction of the excited state, that is secondary charge separation, isomerization, dissociation, etc. Third is by the non-radiative vibrational quenching of the excitation by solvent. The process re-emission of the radiation by luminescence is of interest in sensing and signaling applications, while chemical reactions are of interest in applications such as molecular switches and photocatalysis. Strictly speaking students, absorption and re-emission type processes are termed molecular photophysics, while light induced chemical reactions or chemical processes are termed as photochemical or photochemistry processes. Supramolecular photochemistry may involve three steps. First one is the binding of substrate and receptor. Second is the mediating of photochemical process such as energy, electron or photon transfer. Third one is it is followed by these two steps. It's either restoration of the initial state for a new cycle or by a chemical reaction. Now this you can see in the figure which is now shown to you. It is representation of photo-induced energy transfer and electron transfer processes involved in supramolecular photochemistry. Now you can see generation of all these types of chemical species may be followed by a chemical reaction. In principle, supramolecular photonic devices require a complex organization and adaptation of the components in space, energy and time, leading to the generation of photosignals by energy transfer or electron transfer, substrate binding, chemical reactions, etc. Now let us focus our attention onto the light conversion and energy transfer devices. A light conversion molecular device may be realized by an absorption energy transfer emission, that is AETE process, in which light absorption by a receptor molecule is followed by intramolecular energy transfer to a bound substrate which then emits. An optimum device may be designed if the light absorber or antenna, light emitter and the degree of coupling between them may be tuned separately. The combination of coupled absorber and emitter thus produces a light conversion device with optimal properties such as maximum light collection and emission at the desired wavelength. This occurs in europium-3 and terbium-3+ cryptates of the macrobicyclic ligand bipyridine complex. UV light absorbed by the bipyridine groups is transferred to the lanthanide cation, for example europium and terbium-3+, bound in the molecular cavity and released in the form of visible lanthanoid emission via the AETE process. The europium-3+ and terbium-3+ complexes display a bright luminescence in aqueous solutions, whereas the free ions do not emit anything under the same conditions. Now you can see this figure where it is actually showing you the AETE process is shown with the following, that is europium-3 tris bipyridine cryptate complex. Now after having studied this topic, let us concentrate on the photo-induced electron transfer in photoactive devices. The photogeneration of the charged separated states by photo-induced electron transfer, also known as PET, is of interest for initiating photocatalytic reactions. For example, natural and artificial photosynthesis, and for the transfer of photosignals through a membrane, for example. A schematic diagram of a simple example of such processes can be seen in the figure. Here in this figure you can see a 3-component entity containing a photosensitizer, that is PS, linked to an electron donor D and an electron acceptor A, possessing suitable redox properties in their ground and excited states, so that irradiation of PS, that is the photosensitizer, leads to electron transfer from D to A, that is donor to acceptor, yielding the charge separated triad D plus PS A minus, that is the donor with the loss of electron, the photosensitizer, and the acceptor with the gaining of electron. Now here again this figure is clearly showing you the schematic representation of the process discussed earlier. An example of a photo-induced charge separation is shown to you in the figure. In this, a macropolycyclic coreceptor containing both a photosensitive porphyrin ring and binding sites for silver 1 ions as acceptor centers. This results in quenching of the singlet excited state of the zinc porphyrin center by an efficient intracomplex transfer, leading therefore to charge separation and generating a porphyrinium cation for a long lifetime. Now here you can see the same, The structure is beautifully shown to you here. That is a microcyclic zinc porphyrin co-receptors containing both the photosensitive porphyrin group and the binding sites for the silver 1 cations as the acceptor centers. Now students, let us focus our attention now to molecular and supramolecular electronic devices. Molecular electronic devices conceptually resemble electronic and computer components. If individual electronic components are coupled together, such as wires, switches, rectifiers, etc., they can be produced. Then the conceptual basis exists to manufacturing molecule-sized, that is nanoscale electronic devices with concomitant gains in speed, efficiency, capacity and reduced use of resources. Much attention has been given to design electronic devices that would operate at the level of molecular and the supramolecular level also. Very extensive work has been performed on electroactive materials such as organic metals, for example, conducting polymers and charge transfer salts, or molecular semiconductors that may lead to devices based on organic materials. This defines a field of molecular and supramolecular electronics concerning the properties of single molecules, of oligomolecular associations and of polymolecular architectures such as Langmuir-Blodgett films within and between which electron transfer processes may occur. The actual realization of such devices involves several steps. It is first necessary to imagine a component molecule that may possess the desired features, synthesize it and study its properties. The second step is to incorporate it into supramolecular architectures such as membranes or other organized structures, and to investigate whether the resulting entity possesses the required properties. The third step requires the connection of the basic unit to other components in order to address it via relay molecules or with an external physical signal. The pursuit of design of molecular electronic circuitry may have many spin-offs along the way, in addition to raising novel questions about the handling of various molecules. Now students, let us come on to the topic of supramolecular electrochemistry. Supramolecular electrochemistry is the resulting electrochemical effects from redox responsive receptor substrate binding and the molecular devices operating on electrons are basic elements for converting molecular recognition events into electronic signals. The redox responsive receptor molecules are based on two types of components: substrate binding sites and electroactive groups. A number of substances have been studied in which the redox properties of groups such as metallocenes, quinones, paraquat are modified on substrate binding. Conversely, redox interconversion of these groups allows the reversible switching between states of high and low affinity. The mutual effects of redox changes and binding strength in a receptor substrate pair thus make it possible to achieve electrocontrol of complexation, and conversely to modify redox properties by binding. As a consequence of these electrochemical effects, molecular receptors may be used to convert the chemical information present in binding and recognition processes into electronic signals. Multinuclear metal complexes possess a rich electrochemistry and may serve as electron reservoirs. Thus, dinuclear copper-1 and nickel-2 complexes exchange 2 electrons in one step and 4 electrons in 2 steps respectively. Attachment of several redox groups such as ferrocene viologen or nickel-2 cyclam to a core provides electron reservoirs that may be capable of powering multi-electron catalytic reactions. Polyoxymetallates also take up a number of electrons, up to 32 electrons. Biological multi-redox centers are found, for instance, in the electron transfer protein cytochrome C3 that contains 4 heme groups whose redox properties are affected by local solvation, as in the synthetic porphyrins. Now students, let us discuss, after having discussed the topic of synthetic porphyrins and the other devices, let us discuss the electron conducting devices, that is molecular wires. The basic properties of a molecular wire are that it should connect to two components, generally an electron acceptor and an electron donor, and conduct an electrical signal or impulse between them. The three main classes of electron transfer processes may be considered. First one would be the electron transport by redox active molecules acting as mobile carriers through membranes. This may or may not also involve electron transfer from one carrier to another during an encounter. Second would be electron hopping between suitable redox active groups attached to a backbone or assembled via non-covalent interactions. Third is the electron conduction along a continuous conjugation path formed by pi bonds, which may also involve other electron transmitting groups such as strained sigma bonds, etc. It represents an electron channel and embodies specifically the idea of a molecular wire. Thus, the molecular wire concept is analogous to the channel mediated mechanism, since it is expected to involve a rapid current flow, but the ideal electronic device would result solely in electron flow rather than the flow of alkali metal cations. Now this figure here shows you the triplet excitation transfer between ruthenium-2 donor and osmium-2 acceptor in this particular complex. The design of a molecular wire should satisfy three basic criteria. First one is it should contain an electron conducting chain. Second, it should possess terminal electroactive and polar groups for reversible electronic exchange. Third is it must be long enough to span a typical molecular supporting element such as monolayer or bilayer membrane. Here, transmembrane electron transfer processes are shown. You can see that carrier mediated via a redox carrier, which is on the left, or a channel mediated via a molecular wire, which is represented on the right. Both processes may be coupled to light by introduction of photoactive groups in the carrier or in the wire. Electron transfer occurs between an external reducing phase and an internal oxidizing phase on incorporation of zwitterionic caroviologens, that is Cv2 plus minus. This is represented as Cv2 plus minus, that is the zwitterionic caroviologens, because the zwitterion has both negative and positive charges in equal amount, and this compound into phospholipid vesicles. Using only small amount of incorporated Cv2 plus minus, that is the caroviologens, about 150 active molecules per vesicle, an acceleration factor of 4 to 8 over background was obtained. This clearly indicates that these caroviologens, the zwitterionic ones, did induce electron conduction. Now this is shown to you in this figure. Here it is a schematic representation of a transmembrane electron transfer from a reducing agent such as sodium dithionate to an oxidizing agent such as potassium ferricyanide by a zwitterionic caroviologen incorporated in the bilayer membrane of a phospholipid vesicle. Let us now discuss the molecular and supramolecular ionic devices now. The numerous receptor, reagent and carrier molecules are potential components of molecular and supramolecular ionic devices that are capable of handling inorganic and organic ions via highly selective recognition, reaction and transport processes with coupling to external factors and regulation. Such components and the devices that may be built up to form the basis of the field of molecular ionics. The field of systems operating with positively or negatively charged ionic species as support for signal and information storage, processing and transfer. In view of the size and mass of ions, ionic devices may be expected to perform more slowly than the electronic devices. However, ions have a high or very high information content by virtue of their multiple molecular or supramolecular. Molecular would include charge, size, shape and structure, and supramolecular features will include binding geometry, strength and selectivity. So these are the features which make the ions have a high information content. The molecular recognition events involved may be directly related to information and signal processing by ions as in the case of biology. Indeed, biological signals and communication events are based on ionic and molecular species, that is sodium, potassium, calcium, chloride, acetylcholine ions, etc. Selective ion receptors represent basic units for ionic transmitters or detectors. Selective ion carriers correspond to ionic transducers. These units may be fitted with triggers and switches sensitive to external physical, that is light, electricity, heat and pressure, or chemical other binding species regulating site stimuli for connection and activation. Binding, transport and triggering may be performed by separate species, each having a specific function as in multiple carrier transport systems. This allows a variety of combinations between photo and electroactive components and different receptors or carriers. Light and redox sensitive groups incorporated into receptors and carriers affect binding and transport properties. Co-receptors and co-carriers provide means of regulation via cofactors, co-bound species that modulate the interaction with the substrate. Thus, a simple ionizable group such as carboxylic acid functional group represents a proton switch and leads to proton gated receptors and carriers responding to pH changes, as seen for instance in the regulation of transport selectivity by a lipophilic carrier. This is showing the containing a single cation receptor site and two ionizable carboxylic acid groups was found to transport selectively the calcium 2+ ions in the dicarboxylate form and potassium ions when monoionized, thus allowing pH control of the process. This striking change in transport features as a function of pH involves pH regulation of calcium-potassium selectivity in a competitive symport coupled to calcium proton and potassium proton antiport in a pH gradient which provides a proton pump. Now this can be seen clearly in this figure. Ion transfer takes place through mobile carriers or ion channels. A rotating shuttle process may be also considered. Artificial transmembrane channels have been much less explored than carriers, probably because of the inherently larger molecular structures involved, despite the fact that biological transport is thought to occur mainly via channel type species. Various routes to the design of ion channels and ion responsive membrane systems have been investigated. One may imagine a variety of ion channel types depending on the nature of ion binding sites, on the way in which they are arranged, on the molecular type and on the overall structural features. As seen, you can see the next figures. First of all, this figure is showing you the transport processes and the other one is showing you the schematic representation of different possible types of ion channel structures. Now students, after having discussed different types of devices and the concept of semiochemistry, let us summarize this module. In this module, you have learned that the understanding of the design and properties of molecular and supramolecular devices as basis for the development of chemical information processing and signaling, as well as for the exploration of their relationships with related biological phenomena. The development of circuitry and functional materials at the nanometer scale by the formation of photonic, electronic, ionic switching devices from molecular components and their incorporation into well-defined organized assemblies. A supramolecular device comprising two or more components linked either covalently or non-covalently with distinct functions that are properties of individual components. Supramolecular sensors comprise binding, transduction and actuator components either in a single molecule or as a part of an indicator displacement assay. Molecules can display characteristics of electronic components such as conduction of energy or electrons.